partner Caroline Dwarnicka, and today we'll be explaining why biologists initially thought that proteins were likely carriers of genetic information. Okay, so long before biologists discovered structure of DNA, they understood the concept that inherited traits and genes were associated with chromosomes. Chromosomes were discovered in the 19th century in the nucleus of eukaryotic cells when the cells were undergoing cell division. And chromosomes were named after the word chroma, meaning color, because of their staining properties.
In 1928, a bacteriologist by the name of Frederick Griffith conducted experiments using bacteria and mice. He was trying to develop a vaccine against pneumonia but discovered living virulent bacteria in mice. This bacteria had a polysaccharide coat. Polysaccharides are chains of carbohydrate molecules. He discovered the transforming principle, which is given to the substance that could be transferred from non-living to living cells.
Currently, we know DNA carries genetic information, and the protein components of chromosomes function largely to package and control. However, in the 1940s, especially the early 1940s, biologists had trouble justifying accepting DNA as genetic material because of the simplistic chemistry that they had known about. DNA is simply a long polymer composed of nucleotide subunits. DNA was considered too simple of a molecule to hold genetic/complex information. Proteins showed more variation because of their diverse amino acid sequence, and DNA was thought to act as structural support for chromosomes.
Biochemical analysis was developing. Researchers learned that DNA and protein was contained in chromosomes. However, researchers did not know if DNA or proteins were responsible for encoding the genetic information. Later in 1944, researchers Oswald Avery, Macklin McCarthy, and Colin McLeod did several experiments that led them to thinking that DNA was the carrier of genetic material, but realized from their results that it was not. They showed that DNA can transform the properties of cells, clarifying the chemical nature of genes.
It was not until 1952 when Alfred Hershey and Martha Chase used a different approach and identified DNA as the genetic material. After integrating this into either protein or DNA, they recognized that DNA was the only material transferred directly from bacteriophages into bacteria.
Hershey and Chase used bacteriophages to track the transfer of proteins and DNA between a virus and its host, which led to their discovery. Bacteriophages are viruses that infect the bacteria. They are composed of protein and DNA.
Then, in the early 1950s, Maurice Wilkins and Rosalind Franklin used X-ray diffraction to analyze the 3D structure of DNA. In 1953, the results of Wilkins and Franklin's discoveries proved very useful to Watson and Crick's double helix structure.
The structure in which two strands of DNA are wound around each other in helix form suggested how DNA could encode genetic information.